Hi learners, I am Dr. Gurunathan from Jayanti College, and today we are going to discuss about bacterial growth curve, a very important topic and a very interesting one too. To begin with, we are going to discuss about what is a bacterial growth. Basically, it represents increase in the number of bacterial cells rather than increase in their cell size. And usually the growth of bacteria occurs in an exponential manner because binary fission process is by means of which the bacteria is actually growing. And in this, one cell divides into two, then it divides into four, then it increases in number to 8, 16, 32, and the process continues in an infinite stage. And the time taken for a bacterial cell to become doubling, right? Okay, is called as generation time, also called doubling time. And there has been different organisms with a different doubling time. Clostridium perfringens has 10 minutes, which is the fastest one. And Escherichia coli, which is the model organism that is being employed, has 20 minutes of doubling time, whereas the slowest in terms of doubling time is Mycobacterium tuberculosis, which takes around 12 to 16 hours of one cell becoming into two cells. And when you're speaking about the growth curve of an organism, a bacteria shows a predictable growth pattern, particularly when it is allowed to grow in a closed system. For example, it can be a test tube which has nutrients in them or nutrient medium in them, or it can be a fermentar kind of setup where it contains enough nutrients for its survival. And this predictable growth pattern is commonly represented with a term called bacterial growth curve, and it is depicted as a graph that shows how the quantity of bacteria, particularly their numbers, changes over a time period. As bacteria reproduce by binary fission, the growth can be plotted as the logarithm of the number of viable cells against the time of incubation. And there are four distinct phases observed in bacterial growth curve following the inoculation into a new nutrient medium. The first phase is called as the lag phase. The second one is called as the log phase. The third one is called as a stationary phase. And the fourth phase is called as the death phase. Let us talk about the lag phase, which is also referred as a lag stage. Once the organism is introduced into a new nutrient medium, it will take some time to adapt to the new environment. In this first phase, the bacteria does not reproduce but produces or prepares itself for a reproduction. The cells at this stage are metabolically active and they keep increasing in size. They also synthesize RNA, growth factors, and other molecules required for their cell division. Because it is involved in preparing the cells for the next stage, it is often referred as preparation phase or preparation stage, and it varies considerably in length depending on the nature of the medium and the microorganism that is multiplying or that is growing in the medium. When you see this picture, the left hand corner, right, which is circled there, indicates the lag phase. And you can see that there are only few live cells in this phase, and the organisms that are present there, they are actually not multiplying, but they are actually growing in their size. And they are preparing themselves so that they can move on to the next stage or the next phase of the that is called as log phase or log stage. In the second phase of the bacterial growth curve, you see that the cells are undergoing a rapid multiplication, and the cells enter the log phase immediately after the lag phase, and because there is marked doubling of the bacterial cells in a logarithmic fashion, it is often referred as exponential phase or exponential stage. Particularly when you're going to have organisms in a tube of culture medium can maintain logarithmic growth only for a limited time period. And this phase continues until there is depletion of nutrients in the setup where the organism is growing. The stage also comes to a stop if toxic substances are accumulating, resulting in slower growth rate. The cells are at the healthiest at this stage, and because of which several researchers prefer to use bacteria from this stage for their experimental processes. And the exponential phase is actually shown here. As you can see here, there is a gradual increase in the organism, particularly the live cells doubling very fast. And it is indicated in the form of a curve, right? Okay, as you can see there in both the pictures, in the bottom picture it is circled, whereas in the top picture it is shown as such for easiness of understanding. And when you speak about the third phase, it is called as the stationary phase or the stationary stage. And this stage occurs because during the log phase, the nutrients are used up, the metabolic waste accumulate, the living space becomes a limiting factor for the organisms, and the aerobes suffer from oxygen depletion. All these things affects growth rate of the bacterial cells, and because of which now the cells enter stationary phase. As the name says, in this phase the cells are undergoing, or the rate of growth of cells becomes equal to its rate of death, and because of which the cell population remains constant, and that makes this stage as stationary phase or a stationary stage. An additional point that we should consider is if you are working on secondary metabolites of the organisms, then this will be the ideal stage for us to work upon. So secondary metabolites are produced during this stage, and we can definitely use cells at this stage to study any interesting secondary metabolites that they will produce among themselves. And as you can see there in the picture, the stationary phase is just a horizontal line which is visible there, and you can see that it has many live cells. At the same time, the number of dead cells are also increasing equally. That makes it a steady phase. And the last phase is the death phase. In this death phase, as you can see here, there is a decline in the number of viable cells happening, and because of the decline in the number of viable cells, it is often referred as a decline phase, and the decline takes place in a logarithmic way, very similar to your log phase. That is the second phase of your bacterial growth curve, and because of this it is indicated by the straight downward sloping diagonal line in the graph that is usually displaying or depicting the bacterial growth curve. And duration of this phase is as highly variable as your log phase. And the picture here depicts the death phase, the decline phase in the form of dotted lines in the top picture, and it is for easiness of understanding is circularly represented. A circular, red circular representation is shown for the death phase as far as we are concerned. To summarize what we have just seen, as you can see here, this is a simple bacterial growth curve which represents you the lag phase, which is the first page, which is followed by the log phase and the stationary phase and the death phase. And there each of their importance is shown here. And thank you for your listening. We'll meet in another interesting video soon. Take care and bye.